Have you ever picked up a prescription and just wondered how it came to be? I mean, the journey from a scientist's initial idea all the way to that little bottle in your hand is honestly one of the most complex, expensive, and time-consuming processes in all of modern science. So today, let's pull back the curtain and really dig into this incredible journey.
It all boils down to this one simple question, right? What does it really take to make one new pill? Well, the answer is kind of staggering. We're talking about a journey that can take more than a decade, with odds of success that are, frankly, almost unbelievable. But before we even get into the crazy how of it all, we got to understand the why. This whole massive undertaking isn't just some scientific puzzle. It's a direct response to very real, very urgent human needs.
You know, this whole gigantic process really gets started for one of three main reasons. First, for diseases where we have nothing, no effective treatments at all. Second, to find safer alternatives for drugs we already have that might have some tough side effects. Or third, just to create medicines that flat out work better than what's currently available. Every single one of these paths starts with the exact same goal: improving and, hopefully, saving lives.
Okay, on to the first major phase. So, once scientists know which disease they're going after, they need to find a weapon to fight it. And this is the ultimate search for a needle in a haystack the size of a mountain, really. So, scientists don't just start testing chemicals at random. That would be impossible. The very first, crucial step is called target identification. They have to pinpoint a very specific little piece of our biology, maybe an enzyme or a receptor, that's directly involved in the disease. This gives them a precise target to aim for. And the bullets, so to speak, that they design for that target, well, they can vary wildly. As you can see here, they might be tiny little small molecules or slightly bigger short peptides, or even these huge, incredibly complex structures we call biologics. To find the right bullet, they use some absolutely incredible technology. You've got high-throughput screening, which uses robotics to physically test thousands and thousands of compounds all at once. Then there's virtual screening and molecular docking, which are kind of like super-powered computer simulations, predicting how different molecules might fit into the target. And now, of course, AI and machine learning are completely changing the game, predicting promising candidates and just dramatically speeding up that search. Now, what's really fascinating is that even when they find a promising lead compound, it's almost never perfect right out of the gate. So it enters this phase called lead optimization, where chemists basically get in there and tweak and modify its structure to make it safer, more stable, and ultimately a much more effective potential drug. So, a promising candidate has been found, it's been optimized, but now it has to enter the gauntlet. This is a series of absolutely grueling tests designed to eliminate the vast majority of contenders. This slide right here, this shows one of the most important concepts in all of drug development: the funnel. Scientists might start with, say, 10,000 promising compounds, but as they move through testing, that number just gets whittled down again and again. It's a process that is literally built on failure, all to find the one that succeeds. The first big filter is preclinical testing. Before a potential drug ever goes into a person, it is tested extensively in the lab, that's in vitro, and in animal models, that's in vivo. This is the absolute critical safety check to see if the drug works, and just as important, to see if it's toxic. Okay, so if a drug manages to pass all those preclinical tests, it can finally move into human clinical trials. And you can really think of these three phases as just answering three very simple but incredibly vital questions. First, is it safe? Second, does it actually work? And third, is it any better than what we already have? First up is phase 1. This part is all about answering one question and one question only. Is it safe? Here, a very small group, maybe 20 to 100 healthy volunteers, takes the drug. The only goal here is to find a safe dosage range and just watch for any immediate side effects. So once safety is confirmed, the trial gets bigger and moves into phase 2. Now the group is larger, up to 500 patients who actually have the disease, and the focus shifts to that second big question: does it work? This is the very first time scientists get to see if the drug has any real effect on the illness itself. And finally, we hit the massive scale of phase 3. This is by far the largest, longest, and most expensive phase, involving thousands of patients. The goal here is to nail down that third question: is it actually better? You need definitive, statistically significant data to prove the drug is effective and safe in a huge, diverse population before it can ever even be considered for approval.
So when you add all of this up, the timeline is just mind-boggling. From that initial spark of an idea, through years of lab work, all 3 phases of clinical trials, all the way to the final approval, you are looking at an average of 12 to 15 years. Think about that, a decade and a half.
But that simple linear timeline, it's a little deceptive. This chart brilliantly illustrates the true complexity. It is not one team doing one thing at a time. No way. It's a massive parallel effort, coordinating chemistry, toxicology, manufacturing, and legal teams all at once. It's a logistical masterpiece, really, where just one delay in one area can cause a cascade of problems for the entire project.
So what happens when a drug candidate has somehow survived this entire decade-long gauntlet? Well, it finally approaches the finish line.
This is like the final exam. All of the data, from every single lab test, every preclinical study, and every clinical trial is bundled up into something called a new drug application, or NDA. That gets submitted to regulators like the FDA, and they pore over every single page to decide if the drug should be approved.
But you know what? Even after approval, the work still isn't over. This brings us to phase 4. After a drug is on the market, it is continuously monitored for long-term safety out in the general population. This is to make sure that any really rare side effects that didn't pop up in the trials get caught.
Okay, let's zoom all the way back out now and put this entire journey into perspective, because the final numbers, they really show the true scale of this challenge.
Let's look at that funnel one last time. Just think about it. The years of work, the millions, sometimes billions of dollars spent on those thousands of initial compounds just to find 5 that are promising enough to even begin human trials. The sheer scale of failure is just baked into the system. And of those 5 candidates that make it into trials, after more than a decade of work, after an investment that can easily exceed $1 billion, and after the dedication of thousands of patients and researchers, you are often left with just one single approved drug. And that leaves us with a final and pretty crucial question. The fact that we can succeed at all is just a testament to human ingenuity. But with odds this low and costs this high, the challenge is clear. How are we going to innovate to make this process faster, cheaper, and more successful? How will we find the cures we so desperately need for tomorrow?